Our fMRI tutorial series concludes with the last major software package, SPM. SPM stands for Statistical Parametric Mapping, which is the method of mapping your imaging results onto a brain. The finished product looks like those images you see in magazines that show you what is activated when watching a movie, or what part of the brain lights up when you take a poop. So why use SPM? Among all of the other major software packages, there are trade-offs between flexibility and how easy it is to use. For example, AFNI is very flexible, but has a steep learning curve. SPM, on the other hand, can do analyses entirely from a graphical user interface, or GUI. You can learn the basics by clicking a few buttons, and the user is guided through each of the analysis steps. All of this comes at the cost of reduced flexibility. What you see in the GUI is what you have control over. There are some options that are available if you use the commands from the SPM library, although they can be difficult to use if you don't have experience with coding. As we learn more about the basics of SPM, we'll eventually learn how to code our analyses or script them in order to save time and minimize the chances of making a mistake. Another feature of SPM is that it is run through MATLAB. This is an advantage for some people since they can use any operating system they want, in contrast to AFNI and FSL, which require Unix or a Unix emulator. For others, MATLAB is both an expensive software package and an awkward programming language. But if you're part of a laboratory or university that has access to MATLAB for free, it may be worth your time to use it. So how do you get SPM? Assuming that you have already downloaded and installed MATLAB, you should first go to the SPM12 website and click on the Download Form button. Select SPM12 as the version that you want, and fill out the rest of the details. Once you've filled out the rest of the options, download the software package by clicking the Download button down below. This may take a few minutes, so we're going to fade out while it downloads. When it has finished downloading, click on the zip folder to unzip it, and then open up Finder and move it to your home directory. You can find your home directory by clicking on Go at the top of your screen. Once you have finished moving it to your home directory, open up MATLAB, then click on the Home tab, and then click the Set Path button. From this screen, select the SPM12 directory in your home folder, and then click Open. And then Save to automatically set the path each time MATLAB is opened, and then close the window. After you have set the path, type the following from the MATLAB terminal, SPM, and press Enter, which opens up the following window, and then click on the fMRI button to open up the SPM fMRI graphical user interface. When you're finished with it, it should look something like this. Now that you've downloaded the dataset and learned about the flanker task, you'll want to inspect it for any problems. As you gain more experience looking at these images, you'll be able to judge whether there are minor flaws that can be fixed by pre-processing, or whether there are major issues that indicate a problem with the scanner. In either case, you will need to look at the data carefully by using the SPM viewer. Let's first rename the dataset to something that is clear and informative. If the dataset has been downloaded to your Downloads directory, first unzip it, and then open a MATLAB terminal and type the following, which will rename the folder to Flanker and put it on your desktop. As you saw in the previous tutorial, the dataset has a standardized structure. Each subject folder contains an anatomical directory and a functional directory labeled anat and func, and these in turn contain the anatomical and functional images collected during the experiment. This format is known as BIDS, or Brain Imaging Data Structure, which makes it easy to organize and analyze your data. To begin, let's take a look at the anatomical image in the anat folder for sub-08. Navigate to the sub-08 folder and then the anat directory. Before we can look at the images in SPM, we will first need to unzip them using the gunzip command. Type gunzip *.gz to unzip everything in the directory, and then navigate to the func directory and type the same command. Then navigate up one directory and type spm fMRI. If you click on the display button, you will be prompted to select an image. Select the anatomical image sub08t1w.nii. When you click on the Done button, the image will be displayed in a new window. Inspect the image by clicking around in one of the viewing panes. Notice how the other viewing windows and crosshairs change as a result. This is because MRI data is collected as a three-dimensional image, and moving along one of the dimensions will change the other windows as well. You may notice that this image doesn't have a face, which has been deliberately removed in order to ensure the subject's anonymity. If you were to observe that other parts of the brain were missing, however, or that there were distortions in the image, in other words, if you saw artifacts, you should make a note of this. Some are due to bad data quality or movement, or because of scanner malfunctions. Click on the link below for examples of artifacts to look out for. When you are done looking at the anatomical image, click on the display button again. Navigate to the func directory and select the Run1 functional image. A new image will be displayed in the orthogonal viewing windows. This image also looks like a brain, but is not as clearly defined as the anatomical image. This is because the resolution is lower. It is typical for a study to collect a high-resolution T1 weighted or anatomical image and lower resolution functional images, which are lower resolution in part because they are collected at a very fast rate. One of the trade-offs in imaging research is between spatial resolution and temporal resolution. Images collected at higher temporal resolution will have lower spatial resolution, and vice versa. Many of the quality checks for the functional image are the same as with the anatomical image. Watch out for extremely bright or extremely dark spots in the gray or the white matter, as well as for image distortions such as abnormal stretching or warping. One place where it is common to see a little bit of distortion is in the orbital frontal part of the brain, just above the eyeballs. There are ways to reduce this distortion, but for now, we will ignore it. Another quality check is to make sure there isn't excessive motion. Functional images are often collected as a time series, that is, multiple volumes are concatenated together into a single dataset. To view the time series of volumes in rapid succession, right-click on any of the planes and click the Browse button. You will be prompted to select an image. Click on the currently selected file to remove it, and then enter the string run-1 in the filter field, and 1:146 in the frames field. Select all of the resulting images by right-clicking and clicking on Select All, and then click Done. You will now see a horizontal scrolling bar at the bottom of the display window. Clicking on the right or left arrows will advance or go back one volume. You can also click and drag the scrolling bar to view volumes more rapidly. Clicking on the right arrow button in the bottom right will start movie mode, which flips through the volumes at a rapid pace. Clicking on the button again will stop the movie. To see a plot of the time series activation at the voxel under the crosshairs, right-click again on any of the planes and select Browse, and then select Display Profile. This opens up another figure that you can view simultaneously as you flip through the volumes. Earlier, we learned that the goal of this course is to test for differences in brain activity between two experimental conditions, incongruent and congruent flanker trials. We expect that brain regions involved in cognitive control will be more active in the incongruent condition compared to the congruent condition. In order to test this, we'll first need to pre-process our data, which removes artifacts and cleans the images. If you've ever taken a picture and found problems with it, blurriness, red eye, or faded colors, for example, you can use an image editor to remove those problems. We do the exact same thing with three-dimensional images of the brain taken with the scanner. Each of our pre-processing steps will clean up the images and prepare them for statistical analysis, which we will discuss in a later video. To begin, open a MATLAB terminal, make sure SPM is open, and then navigate to the sub-08 folder within the Flanker directory. Our first pre-processing step is realignment, which will align all the individual volumes together like cards in a deck. If you click on the button Realign, Estimate, and Reslice, a window opens up showing the options for realigning and reslicing the data. The estimate part refers to estimating the amount that each volume is out of alignment with regards to a reference volume, and reslice indicates that the estimates are used to align each volume with respect to the reference volume. The reference volume is set in the field num passes, which allows you to specify whether the volumes will be aligned to the mean of all the volumes or to the first volume. For this tutorial, leave it as the default, and leave the rest of the defaults alone as well. In this experiment, for each subject, there were 2 runs or sessions of data. If you click on the data field, you will see an option to add more sessions. Click on New Session to add another session. You will see an X to the right of each session field, indicating that this field needs to be filled in before the program can be run. Add another session, and then double-click on the first session to open up the image selection window by double-clicking on Session. Navigate to the Func directory and note that there are 2 files within that directory, run1 and run2. The "1" at the end of the file name indicates that only the first frame or volume is available for selection. In order to select all of the volumes for a specified run, we will need to expand the number of frames available for selection. In the frames field, underneath the filter field, type 1:146 and then press enter. However, you will notice that all of the frames for both run 1 and run 2 have been selected, even though we only want the frames for run 1. You could simply click and drag from frame 1 to frame 146 for run 1, but you risk accidentally including other frames by mistake. To restrict our file selection to run 1, we can instead use the filter field. This field uses regular expressions, a type of coding shorthand to indicate which characters to include in a string. In this case, to the left of the dot asterisk characters that are already in the field, type run-1 and press return. This will display only those frames which include the string run-1. Then either click and drag to select all of those images, or right-click in the selection window and click Select All. When you are finished, click Done. Now do the same procedure for the second session using the filter field to restrict your search to frames containing the string run-2. Now that you've filled in all of the fields that have an X next to them, the play button in the top left corner has changed from gray to green. Click on the button to begin the realignment pre-processing step. When the realignment pre-processing has finished, it will generate a motion graph showing how much the subject moved in the X, Y, and Z dimensions. Translations are how much the subject moved along each axis, while rotations show how much they rotated around each axis. A rough guideline is to remove any runs in which the subject moved more than the size of a voxel, in this study more than 3 millimeters, or if there is a sudden spike more than half a voxel size, again in this study that would be 1.5 millimeters. In this study, the subject moved very little, as you can see by the y-axes, and so we will include all of their data. In this video, we will finish the pre-processing module by running all of the other steps, comparing the input and the output of each command to make sure that it ran successfully. The next step is slice timing correction, which edits the data to make it look as though all of the slices were acquired simultaneously. Similar to what we did with realignment, we will first click on the Slice Timing button in the SPM GUI. Click on the Data field and create 2 new sessions. Double-click on the first session, and in the Filter column, type caret r sub -08 underscore task flanker underscore run -1. In the frames field, enter 1:146 and press enter. Select all of the frames that are displayed and click done. Do the same procedure for the run2 files for the second session. Now go back to the batch editor window, double click on number of slices and enter a value of 40, and then click OK. The number of slices can be found using the SPM vol command, which is explained in the link below to the ebook. For the TR, enter 2. For the TA, follow the formula provided in the help window and enter the following expression. 2 minus, in parentheses, 2 divided by 40. For most data sets acquired with a Siemens scanner, the slice order will be ascending and interleaved. Enter this expression in the slice order field. In brackets, 1:2:40, space, 2:2:40, end brackets. And for the reference slice, enter value of 1 to indicate the first slice that is acquired. Leave the file name prefix as is, which will prepend an A to the files that are generated. When you have finished, press the green Go button. This should only take a few moments. As with the realigned data, it is difficult to see any difference with the check reg button, but we will be able to better examine the differences between the input and the output in the other pre-processing steps. Next is co-registration, or aligning the functional data to the anatomical data. Click on Co-register, Estimate, and Reslice. This will open up a batch editor window with only 2 fields that need to be filled in, a reference image and a source image. The reference image is the image that will remain stationary. The source image, on the other hand, is moved around until a best fit is found between the reference image and the source image. For most experiments, you'll want to use a representative of the functional data as the reference image and the anatomical data as the source image, since we generally want to make as few changes as possible to the functional data. Double-click on the reference image and select the mean sub-08 task flanker run1 bold image in the func directory. For the source image, navigate to the anat directory and select the file sub-08 t1w.nai. Then click the green Go button. This step should only take a few moments. When it finishes, another window will be generated showing the co-registration results with the mean functional image on the left and the anatomical image on the right. Click and drag the crosshairs in either image to see how well the images are aligned. In addition to the outlines of the brains being matched, you should also check to make sure that internal structures, such as the ventricles, are aligned as well. Remember that the intensities will be flipped. Darker areas in the anatomical image, such as the ventricles, will appear brighter in the functional image and vice versa. Now that we have co-registered the images, we are ready to normalize all of our data to a standardized space. Before we can do that, however, we need to segment the anatomical image in order to align the different tissue types when we normalize it. Setting up the segmentation step only requires the realigned anatomical file as input. From the segmentation button, double-click the volumes field and select the file r sub08t1w, and then set the Save Bias Corrected field from Save Nothing to Save Bias Corrected. The last step is to change the deformation fields to Forward. This is at the very bottom of the menu. When you have done all of this, click the green Go button. Once the segmentation has finished, you are now ready to use the information generated by this step for normalization. Select Normalize Right, click on the data field in the batch editor, and create a new subject. Select the deformation field that you created in the inat directory, which should be called y_r sub 08 t1w.nii. And for images to write, select all of the realigned and slice time corrected images. You can do this more efficiently by typing !ar dot asterisk in the filter field and entering 1:146 in the frames field. Once the functional images have been normalized, check the output to make sure there were no errors. From the SPM GUI, click on Check Reg and select one of your functional volumes that has a W prepended to it, indicating that it has been warped or normalized. For the second image, go to the directory spm12/canonical and select any of the T1 images, either average 152 T1, average 305 T1, or single subject T1. In this case, I'm going to select the file average 152 T1. As with co-registration, check to make sure that both the outlines of the brains and the internal structures are well aligned. The last step is smoothing, which averages together signal and cancels out noise. Click on the Smooth button and then double-click on Images to Smooth. Select the warped functional images and expand them to include all 146 frames for each run. Leave the other defaults as they are, and then click on the green Go button. As before, use the Check Reg button to load a representative volume from the output you just created, and view it side by side with a warped functional image that hasn't been smoothed. Make sure that it looks that it has been smoothed by the amount that you specified. Now that we've finished pre-processing the data, we are ready to set up a general linear model and fit it to the data, which we'll cover in the next video.